So thank you very much to everyone for joining the Ensembl webinar today. My name is Ben Moore and I am the Ensembl Outreach Project Manager. So I'm in charge of delivering all of the events that are teaching people about how to use the Ensembl genome browser, exploring all the data we have and using it in their research. So today we just have a short webinar where I want to talk to you about how you can use the Ensembl Rapid Release website to explore newly annotated genomes. Before I start, what I wanted to do was introduce you to the materials for the webinar today. We actually have an online training portal for the Ensembl events, where I've actually placed all of the course materials for today. So I'm just going to jump out of the presentation and show you where you can find them. So if you type training.ensembl.org into an internet browser, that will take you to the Ensembl training portal. So this isn't the Ensembl genome browser. This is a separate site that we have with information about all of our training events. And in the very top, if you click on the training materials link in the blue bar, you'll find a calendar of all of our training events. And if you click on the date for today, the 9th of March, with the webinar, you can see that we have all of the information about the course today. So over on the right-hand side, we have a link for a PDF of the presentation slides that I'm about to deliver. And then at the very bottom, we have this section called New Demos and Exercises. So if you click on the module that we have here, and then expand this window, you'll find screenshots of the practical session of the webinar that we're going to go through today. So this is just a description and some annotated screenshots of the different steps that we're going to take as we navigate around the browser. So please feel free to have a look at this and use this as a reference when we're going through the webinar itself. But you can always come back here and use this as a reference in the future as well. So I'm going to put the link for this in the chat box, so you can all access those materials now. Okay, good. So I wanted to start by introducing you to the Ensembl project in general. I'm sure many of you might have come across Ensembl and used it in your work before, but for those of you who are new to Ensembl, I wanted to give you an overview of the Ensembl project. So Ensembl is a genome browser, and genome browsers are really useful because they help us to navigate around a genome, which in the case of human can be up to 3 billion base pairs in size, and for other species even larger. So the first organism to have its genome sequenced was actually a bacteriophage back in 1977. Fred Sanger and his team in the UK sequenced a genome that was just 5 kilobases in size. So by today's standards, that's obviously tiny, but that technology laid the foundations for more and more complex organisms to have their genomes sequenced until in the early 2000s, we had the publication of the first draft of the human genome sequence. Even since then, we have the advent of next generation sequencing technologies, which has meant that more and more organisms around the world are having their genomes sequenced. So we can think of some large global collaborative projects that are aiming to sequence lots of different species. The Darwin Tree of Life project is an example of one of these projects that's aiming to sequence all life in the UK, basically. So all eukaryotic, all eukaryotic life in the UK. And the idea is that these species are having their genome sequenced, and the rate that the genomes are being produced is unprecedented. So even throughout the 2000s and in the 2010s, Ensembl would add a species every few months, and the gene and the annotation process took a long time. But what we're having to do now is provide the resources for hundreds and soon thousands of species that have had their genome sequenced. So when a species has its genome sequenced, this is generally an example of the output that we have. So it's just a string of letters, A's, T's, G's, and C's. And without any context or annotation, this doesn't mean anything to anyone. So this is what Ensembl tries to do. We take the genome sequences from different species, and we annotate them with features that obviously are interesting and important for biological function and for scientific research. So we annotate one of the most important things that we annotate on the sequence itself is the genes. And we think obviously about the genes that are composed of individual transcripts or splice variants. And we think about the biotype and whether they're protein coding or non-coding. And we annotate that all across the genome. We also annotate variation data and also other features as well, such as promoters and enhancers that can regulate gene expression. So as a summary, Ensembl has 4 main types of data. So the first is the genomic assemblies. So it's important to notice that Ensembl is not a sequencing project. So we don't actually produce any of the genome sequences ourselves. We rely on these research groups and collaborative projects to sequence the genomes of different species and then to deposit those genome assemblies into publicly available archives. So once those genome sequences are publicly available, Ensembl imports the genome sequences and uses an automated gene annotation pipeline to predict the gene models across the whole genome. The second type of data is the variation data. So we import both small and large scale sequence variation data for a number of different species where that's available, including things like phenotype associations as well. The third type of data is the comparative genomics. So this is analyses that we perform within the Ensembl team, and it's where we compare the genes and the genome sequences from one species to another. So we produce gene trees to infer homology relationships, and we also perform whole genome alignments as well to look for conserved regions of genome sequence across the genome. The fourth type of data is the regulation data. So this is where we use epigenetic data, DNA methylation data, ChIP-seq data to predict the existence and activity of regulatory features such as promoters and enhancers across the genome. So to draw these 4 branches of data together, Ensembl has a number of features. So firstly, we have what we call our gene build, which is the gene annotation process for it's actually over 300 species now in the main Ensembl website. We also have our variation, comparative genomics, and regulation data displayed in the browser as well. Alongside all of the data that we have there, you can also upload your own data to view alongside the publicly available data. So you can actually visualize your own data sets alongside all of the other data types that we have in Ensembl as well. There are a number of tools that we have for analyzing and processing data. So an example of that would be the variant effect predictor. That's the acronym is VEP, V-E-P. And that's a tool that's used for annotating and interpreting your own sets of genetic variants. You can do all of this through the browser, but there are other ways that you can access the data. So on a larger scale. So we have the Biomart data export tool, which is useful for exporting customized tables of data from Ensembl. And then you can also access the data programmatically through the Perl or the REST APIs. The best thing about this, about all of this, is that it's completely open source. So all of the data we have is completely free for you to take and use and analyze in your work. So as I've said, Ensembl has the genome assemblies and annotation for over 300 different species now. This is just a snapshot of the species that we have at the moment. And it's obviously not important to perhaps look at every individual species, but it's important to notice that the species that we do have in Ensembl are all vertebrate species. So they're all species with a notochord. We obviously have human as the most well-studied species on the planet. And then we also have the other great apes, and then a large number of other primates, and then also placental mammals as well, including agriculturally and conservationally important species as well. We also have a large number of marsupials and monotremes as well, in order to study mammalian evolution. Alongside the mammals, we also have a large number of birds, reptiles, fish, and amphibians in Ensembl as well. So you can access all of the data for the vertebrates through our main Ensembl website. So that's www.ensembl.org. So we've got the vertebrates and important model organisms and all of the other species that we have there, the vertebrates. And then we also have a sister project called Ensembl Genomes. So if you're studying a non-vertebrate species, we have a page for bacteria, for fungi, for protists, for non-vertebrate metazoans, and for plants as well. So if you're studying a non-vertebrate species, then you can navigate to our separate website called ensemblgenomes.org, and that's where you can access all that same data that we've just been describing, but just for the non-vertebrate species. When you're working with Ensembl and Ensembl genomes, it's important to remember that we work with a release cycle. So obviously new species are being sequenced, new data is being generated all the time. So we release a new version of Ensembl every 3 to 4 months. That includes all of the latest data that we've been working on and producing and analyzing. So as an example, in December of last year, 2021, we released Ensembl 105, which is actually the current version of Ensembl that's available when you visit the website at the moment. We're currently in the process of looking at new genome assemblies, updating our, reperforming our analyses, updating our gene sets, updating our web interfaces. And then what we're going to do in the next few weeks is to package that all up and release it as Ensembl 106. So that would be the new version of Ensembl that you would by default visit when you went to www.ensembl.org. Now, this is obviously a very nice process. It's very structured and organized, and you can trace your data through time, and you can see how the annotation and the different data that's come up over the different months and years that you might have been looking at Ensembl. However, this is quite a slow process, and you can imagine how, especially with projects like the Darwin Tree of Life project, when hundreds of species are having their genome sequenced in a very short space of time, this 3 or 4 monthly cycle means that there's data available that isn't accessible through Ensembl, depending on the release cycle. So what we've released in last year, actually, we released this website, is called Ensembl Rapid Release. So this is a lightweight genome browser that has information about species that have recently been sequenced and have been annotated. And we're going to explore the data that's available through the Ensembl Rapid Release site in a moment. So the website is rapid.ensembl.org. So there's a question that's just come in on the chat box, so I can answer this one now before we move on to thinking about the Rapid Release site in more detail. So it says, I'm curious, why was the decision to keep non-vertebrates in a separate site made? The reason is historical mainly. So Ensembl was first sort of launched and developed in the 1999-2000 at the turn of the century. And it was primarily designed as a resource to show, well, to present and make available the human genome sequence and annotation. So over time, we added some more model organisms when they became available, such as mouse and zebrafish. And then Ensembl Genomes was actually only launched in the late 2000s, around 2007, 2008. And they were just separated based on the data that was available and the different species that were available at that time. Obviously, since then, both of the resources have grown and grown and grown. Over the next year or two, maybe 3 years, we're actually planning on releasing a new Ensembl website that actually integrates both of these resources together. So you'll actually visit one Ensembl genome browser that will have both the vertebrates and the non-vertebrates presented side by side. Okay, good. So there's a few more questions that are coming in, but I'm going to proceed with the presentation, and then we'll look at the questions at the end. So we just thought about the rapid release sites, where we have obviously information about these newly sequenced species that have a small amount of data available for them that we've annotated and made available through the rapid release genome browser. So the Ensembl annotation is a very complicated process, and there's lots of data that hangs off the gene annotation. So you can imagine here, for example, this is just a cartoon that shows you perhaps that there's how a gene is annotated in Ensembl, perhaps with 2 different transcripts. And from those transcripts, we calculate other types of data, such as protein domain data. We use our comparative genomics to predict homology relationships. And we also look for links to other databases that have supplementary information about our genes that we've annotated. And from even that data, even more data is sort of produced and made available through the Ensembl genome browser. So we present functions of genes via the gene ontology consortium. So we use the protein domain and homology predictions to present gene ontology information, which gives you information about the function of the genes. From the homology relationships as well, we can also infer phenotypes and gene names. And using the links to other databases as well, we also look at ontologies, variation data, phenotype associations, and gene names as well. So there's a lot of data that sort of grows and grows from the simple gene annotation that we perform initially. However, in the Ensembl Rapid Release website, because of the computation that's required and the number of species involved, we just have a small amount of data that's initially made available for each of these species. So we perform a gene annotation process. So the genes are still annotated in the same way. And then from that, we also annotate the protein domains. And we also have recently begun making homology predictions as well using Diamond. So we do have some information about gene ontology and the gene functions through the Ensembl Rapid Release website that are inferred through the protein domain annotation. So this is the extent of the data really that's available in the Ensembl Rapid Release website. However, if you are studying a species that's presented in the Ensembl Rapid Release website, what you can do is you can actually cross-reference your species of interest with homologs that are represented in the more well-annotated species in the Ensembl website. So that's what I want to show you how to do in the demo. So that's where we finish up with the presentation. We're going to move on to our demo now. So we're going to explore the Ensembl Rapid Release site. We're going to look at the data that's available for the different represented species. And then we're going to look at the recently added homology data and see how we can cross-reference that to more well-annotated species in the Ensembl genome browser. Good, okay. So the first thing, if you want to follow along with, so what I'm going to do now is I'm going to navigate to the Ensembl genome browser. I'm just going to navigate to rapid.ensembl.org. So please feel free to follow along with me. You can either watch and listen, or you can open up a new tab and try to copy what I'm doing and see and explore for yourself as well. So if you want to explore with me, we're obviously going to be using some gene IDs as search terms. So if you want to follow along with me, then I'm going to put the ID of the gene that we're going to search for in the chat box. So please feel free to copy and paste this at the right time, and then we're going to, we can do the demo together. Okay, so in your internet browser, you can visit rapid.ensembl.org, and that will take you to the Ensembl Rapid Release website. So here you can see the main interface. So at the top, we have links to some of the tools that are available. So the main tool that you can use in the Ensembl Rapid Release site is the BLAST functionality. So that might be quite useful to blast against more well-annotated species as well. You can also see links for help and documentation, and also a link to our blog as well. So you can explore the links in the top in your own time. Underneath that, you can see that we have a search bar. So this is a main search bar on the home page that will allow you to search for against all the different species that we have in Ensembl. Although obviously this drop-down list makes it quite tricky when we have over 500 different species presented in the Ensembl Rapid Release website now. So if you scroll down, probably the more useful thing to do is to select your species using the box underneath here. So you can see at the moment we have just 2 highlighted genomes as examples, but there is a link on the left here that says view and download available data for all species. So if you click on this link, okay, that's not good. So if you click on this link, it will take you to a table that shows you all of the different species that are presented in the Ensembl Rapid Release website. So as I said, we have over 500 species represented, or 500 different genome assemblies represented here now. You can see them all in this table here. So what we can begin to do is we can filter our table to search for our species of interest. So for our demo today, we're going to use water buffalo as an example. So we can actually begin typing in the Latin name, which is Bubulus. Which is Bubulus bubulus. And you can see here we have the scientific name, the common name, water buffalo, and then also the assembly accession number. So this is the unique ID of the accession number that refers to the genome assembly itself. You can see that the water buffalo gene annotation was provided by Ensembl. So that's the case for most of the species, although there is also some external groups who have also worked on particular species. And then over on the right-hand side, you can see that we have links to the different files that have information about the gene annotation. So we have FASTA, GTF, and GFF3 files that have information about the gene annotation. We also have a link for the FASTA files that represent the genome sequence. And then we also have BAM files from the transcriptome data, so the RNA-seq data. So if you want to access any of these data files, you can click on the link. For example, if you want to look at the RNA-seq data sets that are available for water buffalo, you can click on BAM, and it will take you to the Ensembl FTP site. So here is a public directory that contains all of the BAM files and also the indexed BAM files, the .baa files that have information, that have data about RNA, that have the RNA-seq data for a number of different water buffalo tissues. And this is going to be data that was integrated through the Ensembl gene annotation as well. So this would all have been used in the gene annotation process. So if we go back to our tab where we were looking at the table of species, if you want to find out more information about a species of interest, you can click on the scientific name in the table. And that will take you to a species-specific homepage. So from here, you can find out a bit of information about the genome assembly. So you can, firstly, you can see the name of the assembly and the accession number again. You can download the sequence just as we did on the previous page. And then over on the right-hand side, you can see information and some statistics about the genome sequence. So you can see here the length of the genome. You can see the date when the gene annotation was started and completed, and then a table showing you the statistics, so the number of genes and transcripts annotated across the genome in total. So what we're going to do is we're going to use the search bar on this page to search for a gene specifically for water buffalo. So I'm going to use my gene ID that I searched for, that I pasted into the chat box earlier. And then we can click go. And from the search results, you can see that we have one entry matching our search term. So this is obviously the ID of the gene that we searched for. And now we can see that we've navigated to the gene tab. So just as the data is laid out in Ensembl, if you're familiar with Ensembl, this page will look very familiar to you. If not, then you can see that we've landed on what we call the gene tab. So in the blue bar at the top, we open up these different tabs that provide us information about the different features that are available. So we've landed in this gene tab, and in the menu on the left-hand side, you can see the data that's available for this gene. So we've landed on a summary page. So you can see on the summary, we have information about the location, so the genomic coordinates and the strand. You can see that this gene has got 2 transcripts annotated. So if you want to find out more information about these transcripts, you can click on Show Transcript Table. And in the table, you can see the IDs of these transcripts. You can see the length of the transcripts in base pairs, the length of the proteins, if they're protein coding transcripts. And so you can basically get more information about the length of the transcripts. Further down the page, you can see that we have a graphical representation of these transcripts. So as well as looking at the length in the table, you can begin to understand the structure of the transcripts by looking at the exon and introns represented here. So the exons are the boxes, and these lines are the introns. Good. So obviously, we won't go through all of the data that's available here, but you can see that we have information about the sequence. So if you want to download the sequence of this gene, you can click on Sequence and export the sequence from this page. In the blue bar at the top, you can see that as well as the Gene tab, we also opened up what we call the Location tab. So if you click on Location, you can see that we're now viewing a page that looks at the genomic location where our gene that we searched for is actually located. So we can see in the main part of the page, we have what we call a chromosomal overview. So this doesn't provide us too much information, but we can see a red box. And this red box is our region of interest. And then the images further down the page focus more and more on this initial region of interest. So in the second image underneath, you can see that this is a 1 megabase overview. So the scale is at the top here. You can see the scale 1 megabase. And we have this red box again. So the red box is our region of interest. And you can see that the data is now arranged into what we call tracks. So we have the contigs track and the genes track in this case. So the contigs track represents the double-stranded DNA. So this blue bar here represents the double-stranded DNA. And the data in all of the other tracks is aligned to the genome. So in the genes track, we can see that the genes are represented as these colored blocks. So we have, for example, this red block here within our red box. This is our gene that we search for, and you can see that the ID is highlighted. So this is our gene of interest. As we scroll down, you can see that we have this what we call region in detail view, which actually shows you the gene in more detail. So it shows you not only the gene represented as a block, but now it shows you the individual transcripts that make up that gene, and it shows you them in there. Yeah, it shows you the exon and the intron structure. If you want to find out more information about the transcripts, you can click on the features and find out more information from the pop-up window and use the links there as well. You can add data to this view if you want to. So using the button on the left-hand side here that says configure this page, you can click on configure this page. From this, in the pop-up window, you can see all of the tracks that are available to add to this view. So there's a number of tracks that you can add, and they're all organized by the category over on the left-hand side here. So we've got tracks that show us information about the sequence and the assembly, tracks that show us information about the genes and the transcripts as well. What I wanted to show you in this demo is the RNA-seq models. So the RNA-seq data aligned to the genome. So you can see that we have 3 different types of track available here. So we've got the BAM files themselves, so showing you the alignment of the RNA-seq data to the genome. And then we have the gene models, so the genes that are actually annotated based on the BAM files for each tissue. So if we just take two tissues as an example, we can use liver and kidney to view these. Oh, where's kidney? Oh, here it is. So to view these tracks, you just click on the box, and then in the pop-up window, check the box to then choose the track style. So you can click expanded with labels, for example. So you can see now that we're going to see the gene models for the RNA-seq data produced from kidney and liver. So when you're happy, you can save and close the pop-up window using the tick in the top right-hand corner. And then that will add these two tracks here that we can see. So the liver RNA-seq gene models and the kidney RNA-seq gene models. So you can begin to see perhaps if there are any particular transcripts or what some people might call isoforms or splice variants that are expressed particularly in one tissue that they're studying, or you can compare tissues from one to the other. Okay. The final thing that I wanted to show you is how you can actually use the data that's present in the Ensembl Rapid Release site to cross-reference against more well-annotated species in Ensembl. So if we navigate back to the Gene tab using the blue bar at the top, we can click on the link in the menu on the left-hand side called Homologs. And this will take us to a page that has a table that shows us all of the predicted homologs that are available in the species in Ensembl. So you can see here, for example, that the water buffalo gene has an ortholog predicted in cow that has a 98% identity and 100% coverage. So this is the gene for the cow in Ensembl. In the main Ensembl site, sorry, in the main Ensembl genome browser. Further down, you can see that we have the human ortholog as well. So this is obviously the human gene that has a lower percentage identity, but while still 100% coverage. So obviously thinking about the evolutionary relationship between water buffalo and cow and water buffalo and human, we can perhaps begin to understand the differences in the percentage identity here. But human is the most well-studied species on the planet, as I mentioned. And when you go to the Ensembl genome browser, you'll notice that human has this wide array of data that's available for the genes that are annotated for human. So if you click on the link here in the table, you can see that we're now navigating to the Ensembl genome browser. So what you might notice, first of all, is that we're not in the main Ensembl website that has a characteristic blue coloring. We're actually in an archive site. So this is an archive for Ensembl 104, which is actually the version of Ensembl that was the current version when the homology data was calculated and predicted. So that's why we link to that specific archive. So this is the ortholog that the water buffalo gene is attached to. So we can see now when we go to Ensembl that we have this much wider array of data that's available. So we can perhaps begin to understand that water buffalo gene that we were originally interested in. So over on the left-hand side, you can see that we have information about the biological function by looking at the gene ontology. So we can begin to look at the biological process. So you can see that this gene is involved in negative regulation of phosphatase activity. If you click on molecular function, you can see that the protein that this gene encodes is involved in phosphatase and protein binding processes. And then in the cellular component, you can see it's in the protein product is found in the cytosol and the cytoplasm. So we can begin to understand perhaps the function of the human gene that's understood and well annotated, and then perhaps extrapolate that to understanding the water buffalo gene as well. You can also find out any associated phenotypes that have been found in human, so phenotypes in the menu on the left-hand side. So you can see that while there are no phenotypes that are associated with this human gene, there are phenotypes that are associated with variants that fall, with human variants that fall within this human gene. So you can see that variants within this gene in human are associated with cholesterol levels, coronary artery disease, metabolism, urate levels in overweight individuals, and some other phenotypes as well. So again, looking at the human gene and the variants and the phenotype associations in the human gene might help you to understand the function and the phenotypes associated with the water buffalo gene. Finally, we can click on external references over on the left-hand side in the menu. And again, here you can find links to get information about expression data. So you can look at the tissue-specific expression patterns from the Expression Atlas portal. You can also go to the websites from HGNC to get information about the gene name. You can find phenotype and disease associations from the MIM gene and MIM morbid databases. And you can also find links to NCBI and WikiGene as well that have information about this SH2D4A gene, which is the name of the gene that you can see at the top of the page. This is the human gene, the ortholog. So that brings me to the end of the demo. Hopefully you found it useful in trying to understand how to cross-reference between Ensembl Rapid Release and the main Ensembl website. Before I finish up with the final few slides, there's just one unanswered question in the Q&A box that I'm going to try and answer now. So it comes from Afira. So it says, I have a query. What if we retrieve data from an old release? Will the retrieved information be applicable for publication? So I think the answer is yes. So generally, so what happens is that obviously a new version of Ensembl is released every 2 to 3 months. So even if you write a paper today using data from the main Ensembl website, in 2 to 3 months, the data that you look at will be, or it could be out of date, or will have been updated on the main Ensembl website. So what people normally do when they're referencing Ensembl data in their publications is to make sure that they also reference a specific release number. So they say, this analysis or this data set was retrieved from Ensembl 105. And what that means is that anyone that's reading your paper can come back to the Ensembl website, look at the archives of Ensembl and see the data that you would have worked with and that you were talking about in your publication. So I think the best thing to do is to always reference the version of Ensembl that you're using when you're referencing any data that you're analyzing. Yeah. So that brings me to the end of the demo. If there are no further questions, what I'd like to quickly do is just present a final few slides just to summarize what we've been covering today. So the Ensembl Rapid Release website is a genome browser which integrates gene annotation and comparative genomics. So they're the two main types of data that we have for the species that are presented in the Rapid Release site. And obviously this is for recently annotated genomes. So genomes that haven't fully been integrated into the main Ensembl website. However, even though there's less data available for these species, cross-referencing to the well-annotated species in Ensembl using the homology relationships can be useful to help build a picture of the genes that you are studying in those less well-annotated species. So if you want any further help and documentation about using Ensembl, there's a number of resources that you can use. So firstly, we have our online courses that are hosted through the EBI training portal. So you can go to ebi.ac.uk/training and find all of the online courses that are available for Ensembl and all the other EBI resources as well. Ensembl also has a YouTube channel. So if you are interested in watching any recorded webinars or tutorials and videos about using Ensembl data and tools, you can access the Ensembl YouTube channel by typing Ensembl training into YouTube. And if you do have any questions or queries about using Ensembl in the future, then you can always email the Ensembl help desk. So helpdesk@ensembl.org is the email address that you can use. And we'll always get back to you trying to help you understand what you're looking at in the browser and helping you with your queries in your work. Ensembl is a completely free resource, but we really appreciate it when people cite us when they use Ensembl data in their work. So you can cite the main Ensembl project using the NAR paper, the annual NAR paper that we release every year. So we have Ensembl 2022, you can obviously reference in your work. And you can also look at those papers and find out about all of the latest data and annotation results that we have there as well. Obviously, we've had a very short webinar today that's focused on a very specific topic. But if you'd like to host a more general workshop at your institute, then we're very happy to organize workshops with individual institutes to help train people about using Ensembl. So if it's something that you think that you'd like to organize for your own institute, then please email helpdesk@ensembl.org and we can arrange a workshop either virtually or now also we're beginning to do face-to-face courses as well around the world. This is the Ensembl team. So you can see here, this is a picture at our last face-to-face retreat in 2019. And this is obviously the whole team. So the data and all of the work that goes into the Ensembl Rapid Release website is actually made possible by all the people that work on the Ensembl project. If you want to follow us, then we have obviously Facebook and Twitter pages and accounts that you can follow if you're interested in keeping up to date with Ensembl via social media. And then we also have a blog as well. So the blog is actually really useful if you think you're going to be using Ensembl a lot, because posts there where we talk about all the latest data that's going to be released in new releases of Ensembl, any upcoming changes, and any announcements about any downtime or maintenance that's going to be occurring in the future. So that brings me to my final slide. So obviously in the list here is just mainly the people that you'd have seen in the photo on the previous slide. And then obviously our funders are listed at the bottom here. So the funders are actually a really important part of the Ensembl project because they provide us with the money and the resources that mean that we can develop Ensembl and make it available and freely available for everyone around the world who needs to use that data. So we're really indebted to our funders as well. So Emma's asking, so are genomes in the rapid release site in progress towards being fully annotated, incorporated into Ensembl, or will they stay in the rapid release site? So the idea is actually that I'm not sure if you were here for this part of the presentation, but someone was asking about the division between Ensembl and Ensembl genomes, so the vertebrates and the non-vertebrates. And I actually explained about how in the next 2 to 3 years, we actually plan on releasing a new Ensembl website that brings these two resources together and actually presents all of the species that we have in Ensembl through one main page. So that's actually going to be the future of Ensembl Rapid Release as well, in that the species don't actually go to Rapid Release before they then move to Ensembl, but they're actually, everything just goes straight into the main Ensembl website. Obviously, that's in a couple of years' time. At the moment, there's not really a plan to promote species from Rapid Release to Ensembl. I think it depends on the species. So, as an example, there are some species that are already in Ensembl, and then new versions of genome assemblies get published as well. So, if chicken, for example, has a particular genome assembly and is represented in Ensembl, we might, and then a new version of that genome assembly is published. The genome assembly might be annotated and placed initially in Rapid Release before it then becomes a new reference that's then represented in the main Ensembl website. So the answer is not 100% clear. I think it's a bit context specific, but that's our general plans for the future.